Mesothelioma is an aggressive type of cancer that develops from the mesothelium, which is a protective lining that covers many of the internal organs of the body. It is commonly caused by exposure to asbestos. Currently there is no cure for the disease and treatment options are limited. However, scientists are actively researching new therapies that may provide hope. One approach that has shown promise in laboratory studies involves the antiparasitic drug fenbendazole. Fenbendazole is commonly used as an oral worming medication for pets like dogs and cats. It works by preventing the formation of microtubules in parasitic worms, disrupting their ability to feed. Interestingly, cancer cells also rely heavily on microtubule formation to divide and spread. Studies have found that fenbendazole may induce cell cycle arrest and apoptosis in various cancer cell lines by inhibiting the polymerization of alpha and beta tubulin. As mesothelioma cells also depend on microtubule formation, scientists hypothesize that fenbendazole may have anti-tumor activity against this cancer. Preclinical research on mesothelioma cell cultures and tumor models seems to support this hypothesis. In vitro experiments have demonstrated that fenbendazole suppresses mesothelioma cell growth and induces cell death in a dose-dependent manner. Importantly, it appears to be selective for cancer cells, exerting minimal toxicity against normal non-malignant cells. Studies in mice bearing human mesothelioma xenografts also found that fenbendazole significantly delays tumor progression and extends survival when administered orally on a daily schedule. Furthermore, combining it with standard chemotherapy drug cisplatin led to enhanced anti-tumor effects, implying it may work synergistically with current therapies. Encouraged by these promising preclinical findings, researchers initiated the first clinical trial of fenbendazole for mesothelioma in 2018. Called the Fenbendazole trial, it is an open-label, single-arm phase I/II trial taking place at the University of Chicago. The primary goal is to evaluate the safety and tolerability of fenbendazole capsules when given continuously to patients with advanced malignant pleural mesothelioma who have progressed on or are intolerant of standard platinum-based chemotherapy. Secondary objectives include determining the maximum tolerated dose, assessing pharmacokinetics, and obtaining preliminary data on anti-tumor activity by measuring target lesion response rate and progression-free survival. A total of 30 to 50 participants will be enrolled in the dose escalation and expansion phases of the trial. Fenbendazole will be administered orally on a 28-day cycle schedule until disease progression or intolerable toxicity. Several dose levels are planned, starting at 500 mg twice daily and increasing up to 1,000 mg twice daily or the maximum tolerated dose. Participants will be monitored closely for any adverse effects. Tumor response will be evaluated periodically by CT or MRI scans, along with regular blood work and physical exams. Safety data has been assessed in the first 16 patients treated at 500 or 750 mg twice daily doses, finding the regimen to be well tolerated without dose-limiting toxicities reported to date. This initial phase I portion of the Fenbendazole trial is projected to continue accruing patients until 2022. Once the recommended phase II dose is established based on safety review, the single-arm expansion phase will provide first insights into possible clinical benefit from fenbendazole monotherapy in mesothelioma. Additional studies may then assess its potential role as part of combination regimens with standard chemotherapy drugs or immunotherapy agents. If efficacy is observed, further investigation into fenbendazole's molecular mechanisms of action and optimal treatment strategies could follow. Overall, the trial offers hope that repositioning this existing veterinary medication may reveal new therapeutic options for mesothelioma patients struggling with limited standard of care options. Continued clinical research is merited based on positive preclinical progress to date. Several in vitro and in vivo studies have provided promising initial evidence that fenbendazole possesses anti-tumor properties against mesothelioma by disrupting microtubule formation. To explore the clinical potential of this repurposing strategy, researchers have launched the ongoing Phase I/II finasteride trial to evaluate fenbendazole capsules as a continuously dosed monotherapy in advanced mesothelioma patients. Preliminary safety results seem favorable, and completing patient accrual will help determine the maximum tolerated dose along with first signs of any anti-tumor activity for this approach. If results remain promising, further clinical investigation is warranted to optimize fenbendazole's incorporation into multimodal mesothelioma treatment regimens. Currently, this trial offers hope that an old antiparasitic drug may provide a new therapeutic weapon against this devastating cancer.